If you've ever felt confused about the complement pathways, natural killer cells, or interferons, then I'll clear that up in this video, so stay tuned. We are going to walk through my micro mind map and I will talk you through the process. And towards the end of the video, I will test your knowledge with a few questions. Now, if it is super confusing or you just need more practice, then feel free to click the link in the description box for your very own Microbiology It workbook. Now, without further ado, let's microbiology it. The complement system is one of the fastest and most efficient tools of innate immunity, and it works by recognizing pathogens and triggering a cascade of protein activations that lead to destruction. In the classical pathway, the process begins when an antibody binds to an antigen that is on the surface of a microbe. This binding allows the complement protein C1 to attach and activate, leading to the cleavage of C2 and C4 into smaller fragments. These fragments form the C3 convertase enzyme, which splits C3 into C3A and C3B. Now, C3B binds to the complex to create C5 convertase, which initiates the assembly of the membrane attack complex, or MAC. Now, the MAC punches pores into the microbial membrane, allowing water to rush in and lyse the cell. Now, this is effective against gram-negative bacteria. Beyond the classical pathway, complement also strengthens immunity through opsonization. When antibodies or C3b coat the surface of pathogens, they act like molecular handles that phagocytes can easily grab onto. Now, neutrophils and macrophages have receptors for both antibodies and C3b. This allows them to bind quickly and engulf the optimized microbe. This is particularly important for gram-positive bacteria, which cannot be lysed by MAC as easily, but can be efficiently cleared through phagocytosis. The complement system also has pathways, two additional pathways that don't rely on antibodies. The first one is the mannose binding lectin pathway that's activated when MBL binds to mannose that's on the surfaces of microbes. The second one is the alternative pathway. It activates nonspecifically when C3b attaches to microbial components like lipopolysaccharides. Now, both of these pathways generate the same key enzymes that lead to C3 and C5 cleavages, now ultimately converging on MAC formation or opsonization. So no matter where activation begins, all complement routes funnel into the same powerful outcome, which is inflammation, phagocytosis, and microbial lysis. Innate antiviral defenses work very differently because viruses hide inside host cells. Now, this makes them harder to detect. Natural killer cells specialize in recognizing cells that are compromised by viral infection or transformation. Now, healthy cells express MHC1 molecules on their surface, which signals to natural killer cells that, I'm a normal cell, I'm healthy, I'm good, do not target me. Now, virus infected or tumor cells often reduce or lose their MHC1 expression to evade T cell detection, but natural killer cells come in and interpret this absence as a danger signal. Now, once activated, natural killer cells release perforin, which are molecules that form pores in the target cell membrane, and then granzymes, these are enzymes that enter through these pores, trigger programmed cell death. Now, this rapid response eliminates and or eliminates the infected cell before the virus has a chance to spread. Interferons are another critical antiviral defense and serve as a communication system between infected and uninfected cells. When a cell detects viral components such as a double stranded RNA, it produces interferon alpha and interferon beta. Now, these molecules diffuse to neighboring cells and switch on that antiviral genes that block viral replication. Now, in essence, interferons create this protective barrier around the affected sites. Now, this limits the spread of viruses even before the adaptive immune system can fully be engaged. Now, viruses with low virulence trigger high interferon production, while more aggressive viruses may try to suppress interferon signaling to gain advantage. Now, together with interferons and the natural killer cells, they form a more powerful early warning and early action system that helps, contain the viral infections during the critical first hours and days.